My name is Aurora Toennisson, and today I'm going to talk about my NC Space Grant project, investigating plant light and gravity sensing in simulated microgravity. Gravitropism is directional plant growth in response to gravity. Phototropism is directional plant growth in response to light. These tropisms are critical for plants to access the light and water they need to survive. Unfortunately, on Earth, where gravity is constant, it can be difficult to separate triggers of gravitropic and phototropic response. The microgravity environment of spaceflight allows for decoupling of light from gravity cues and makes space a prime place to study plant tropisms. Hardware with rotating centrifuges, such as the European Modular Cultivation System, can simulate Earth gravity and provide 1G control in the spaceflight environment. Studies of plant tropisms will lead to better understanding of how plants work and also point to genes that are important for the cultivation of plants during spaceflight. Plants will be critical to long-duration space missions for providing supplemental nutrition, psychological benefits, and biological life support for astronauts. Others in my lab previously performed 2 independent experiments on the International Space Station, where gene expression in Arabidopsis plants was compared between microgravity-grown plants and plants grown in a centrifuge that simulated 1G Earth gravity. These 2 experiments had significant overlap in upregulated genes. My project is to further investigate the role that these genes play in light and gravity sensing. I'm focusing in particular on genes known to be related to auxin and light signaling. Auxin is a plant growth hormone that promotes cell elongation and curvature of roots and stems. To mimic microgravity, I grew plants on rotating devices called clinostats. A 2D clinostat rotates along one axis. A 3D clinostat rotates along 2 axes. I also grew 1G stationary plants under the same conditions as the control treatment. I extracted RNA from these plants and compared gene expression of the simulated microgravity plants to the 1G plants using a technique called RT-qPCR. This graph shows results from my first experiment growing plants on the 2D clinostat for 24 hours. The 1G reference gene on these types of graph are always normalized to 1. Bars that are above 1 indicate higher gene expression, and bars that are below 1 have lower gene expression. For this experiment, 2 genes, indicated by these asterisks, were significantly upregulated, and another was close to significant, but not quite. However, when I repeated this experiment on the 3D clinostat, I didn't see the same upregulation after 24 hours of growth or after 7 days of growth. In short, alteration of gene expression depended on the duration of rotation and type of clinostat, and our current setup may need to be optimized to better mimic spaceflight conditions. I'm also running a series of experiments of mutants that have genes of interest knocked out so they no longer function. I'm testing if these mutants have altered gravitropic or phototropic response. For example, in my phototropism experiments, I changed the direction of light to come from the side of the plate, and then record a time series of images of how the plants bend. These experiments are ongoing, but so far, at least 1 mutant line shows differences in response. I plan next to engineer plants with luciferase reporters to further investigate the timing of these genes' expression. These plants will contain a firefly gene that will cause them to glow slightly when the gene of interest is expressed. I hope the results of these experiments will improve our understanding of how genes upregulated in microgravity help plants to sense the world here on Earth. I'd like to thank all the people and funding sources that made this research possible, and I'd especially like to thank NC Space Grant for funding my research.